All right, thank you. So I'm going to talk about what I think is one of the biggest pieces of evidence for validity of what Alex has just talked about. We're going to be looking at what do these SNPs actually imply about the way that genes produce gene products and generate brain tissue in different areas of the brain, as well as the actual things that they're doing inside of the cell or whatever. And so we would kind of assume that if these results are not particularly valid because of the crude measures that went into them, that we would, with biological annotation, we would find all kinds of random stuff. We'd find these SNPs expressed in things like skeletal or cardiovascular tissue and so on. But I'm going to try and demonstrate that that is in fact not the case. So just a very brief overview of the kind of methodology that goes into biological annotation. I know that a lot of people are probably not very familiar with this. And so we have a bunch of different tools for doing this nowadays. Most of them have a very cute little acronym. We love cute acronyms in behavior genetics. Some of the most common ones are depict, magma, and more recently FUMA. And I'm not going to really talk about the differences in the algorithms that go into these different tools. They all kind of do the same category of things. They're looking at to what extent are the SNPs from our GWAS expressed or lie near genes or in genes that belong to gene sets that actually do something biologically meaningful. So this is an example. Part of why I'm going to talk about how this kind of thing actually works before I talk about our specific results is just, I think it's incredibly cool. And so this is an illustration I made for Lee et al. 2018, EA3. This shows kind of the last pass of results that we got for this. Let's just start to play. Okay, so these are some of the actual functions that these genes perform in the synapse itself. And so we have things like voltage-gated ion channels, which let calcium, potassium, and so on in and out of the cell. What you just saw animated there was probably my favorite thing, these incredibly cool, elegant little protein machines that are responsible for grasping the vesicle full of neurotransmitters and dragging it across the cell and depositing it against the synaptic membrane, thereby releasing neurotransmitters into the vesicle, which then go on to do other things and pass along signals throughout multiple synapses. And so this is, I just think this stuff is incredibly cool because this isn't just, this isn't really as abstract as it might kind of appear. The things that are happening in this diagram are the things that are responsible for not only a lot of our individual differences in ability and everything else that expresses itself in the brain, but these are the things that are happening when we're literally feeling and thinking about anything. I mean, they're happening just, they're making me speak right now, and you listen, and it's just very cool, right? So biological annotation, you basically just you can use GWAS summary statistics, and these software tools will annotate SNPs in genetic loci. So one kind of pipeline that it can go through is we'll use databases that are external to the summary statistics, and it will build probabilities of association with specific types of tissue. Is it expressed in skeletal tissue or epidermal tissue or hopefully brain tissue in our case? We can also look at these gene set databases such as Reactome and many others to look at what gene sets that are functionally related to some kind of biological process are being expressed associated with our SNPs. And then finally, we can also look at what the specific genes that lie within gene sets that are prioritized by the software, what they're actually doing. What are they producing? You know, genes produce proteins that form machines that do things, right? So the way that we did this project currently is rather than relying solely on depict or magma, we kind of combine them. We use them both separately to annotate SNPs inside of genetic loci, and then both of these tools will kind of filter the ones that pass significance threshold and are also closest to our lead SNPs in the actual GWAS. And then downstream, we can use these external databases to look at what the gene sets are doing and producing, and what kind of tissues are enriched for these genes. And so in our case, we found about 400 prioritized SNPs from depict, almost 18,500 SNPs from magma, but only 94 and 102, respectively, are both genome-wide significant and closest to the lead SNPs that Alex just talked about. And so what we did here is we used FUMA to look at to what extent do these overlap. And if there was nothing meaningful in this data, we probably would find that these two different sets of prioritized genes are not really necessarily related, but in fact we do find massive overlap. 83 of them they come up as being highly significant in both methods. So then we can use FUMA to actually look at functions and different kinds of functions, right? So here's a Manhattan plot of all of the genes, or all of the SNPs that were prioritized, or that were tested for association with magma, excuse me. And we see the ones that are prioritized by both methods in green, and all of them are on the spikes, which we definitely hope to see, and many of them are close to one another. Okay, so first I'll talk about tissue enrichment. What kinds of tissues do these genes seem to be actually producing and enriched for? So what we do is we look at what we call differentially expressed genes. And this is an example from the EA3 that shows that when you use this method, that the kinds of tissues that are prioritized are all brain tissues. And these are, I know they're hard to read down there on that axis, but all of them on the top row here are all brain tissue. And so then I need to talk about what does upregulated and downregulated mean. So upregulated genes are really what we're interested in. These are more highly expressed compared to the reference sample. And so this is going to be the process by which a cell increases the quantity of a cellular component. Downregulated is the functional opposite, that they are perhaps significantly expressed, but in a way that that decreases the quantity of whatever that component is in the tissue. So we're chiefly interested in the upregulated differentially expressed genes. I'm sure the downregulated ones are interesting also, but I'm not really sure how to interpret them, so we haven't really put a lot of thought into it yet, but in any case. Okay, so now I'll talk about some of our results, starting with the two main methods and then the overlap between them with FUMA. And so what we see here is for upregulated differentially expressed genes in the GTEx database, that all of these, pretty much, actually yes, all of them are brain tissue, right? So that's great. That's what we want to see. If we use a different database for tissue, this is the MESH database, then we find something extremely similar, that the brain regions in which, or the regions in which we see the most expression are all brain regions, right? Another way to look at this is we can see what are the actual parts of the brain that these seem to be most enriched for. We see particular expression in the temporal lobe and the occipital lobe, and including here the primary visual cortex and the cingulate gyrus, and a few other things that are kind of, it makes sense that they might be associated with math ability. We also looked at the brainspan development age database to see at what points in an individual's life these genes are most up and down regulated. So you see enrichment here on the y-axis. Obviously, we can't really have negative enrichment. This is just meant to show that downregulation is kind of going in the opposite direction conceptually. And so what we find is that these genes are most expressed around 1 year of age. And somewhat strongly, we have some spikes in prenatal, second and third trimester, and then it kind of peters out to adolescence and adulthood. All right, so I'll briefly now talk about how gene ontology database works. So this kind of gives us multiple levels of analysis, not in tissues, but now in actual gene sets that do things, that do biologically relevant things. So the GEO database considers 3 distinct aspects of how gene functions can be described: molecular functions, cellular components, and biological processes. So these kind of are going from fairly middle resolution to extremely high resolution. And what we find here is, again, this is hard to read, I know, you'll have to just trust me on this, that the enrichment value for all of the significant gene sets that we find here for biological processes, all of them just about have to do with synapses, neurons, and so forth. There's lots of regulation of neuronal processes and activities, type things that show up here, synaptic organization, head and brain development more generally, and many other things that all kind of relate to that. I'll just quickly go over that. We have here, we have like a little heat map that we can make that shows overlapping genes that are within gene sets and the evidence for them playing a role here in our study. And again, you can see that at this bottom cluster here, these things are all brain and synapse and neuron related stuff. Now, if we look at genes, the molecular function part of the gene ontology database, what we see now is that we don't have a huge number of functions that are significant, but all of the ones that are significant are all things that have to do with the synapse and the neuron and synaptic communication, and so on and so forth. And then lastly, we look at the very, very highest resolution thing available to us in this study, which is basically the cellular components of calcium ion binding, in this case, is the one that popped up. And I think that overall, seeing that all of these different things that show up here, these different processes, these different little protein machines that are implicated by the genes that are implicated by the SNPs that we found through this GWAS by subtraction of math ability, that they all have to do with things that involve synaptic communication and brain development and parts of the neuron, and especially the flow of ions in and out of the cell. And so to just kind of briefly close, remember that this is the SNPs that we are using to do this biological annotation. These are net of IQ and years of education, right? So if you think about one of the most brilliant writers that ever lived compared to one of the most brilliant mathematicians that ever lived, what is it about their brains and their genes and the proteins in their synapses that are different from one another if they have the same education and the same IQ? And we do find that even subtracting out years of education and G, that we still have a decent number of things and processes and tissues that are uniquely associated with quantitative ability. So we have vesicle release machinery that is helping to drag that vesicle onto the cleft. We have calcium ion binding, which is responsible for facilitating the flow of calcium in and out of the cell. We have different kinds of receptors that are responsible for neurotransmitter flow, surface receptors, ionotropic, and other kinds of receptors. and mediators of synaptic plasticity, and so on. So these very well may be some of the actual things that are happening at the most basic molecular level that can help us differentiate the Darwins and the von Neumanns among us. So, thank you.
Thank you very much. I very much enjoyed the figures. They're very well done. But I have a question regarding the mapping on the brain regions you showed, because this does partially go against what is known in the literature, especially in the context of dyscalculia, which is the disorder of specific mathematical ability. And usually what is found is that the intraparietal sulcus, or areas around that, seems to be particularly disturbed or inefficient in dyscalculics and seems to be associated with mathematical ability. So the question is, if this is now residualized for G in educational attainment, do you have similar data analogous to this for G to see if actually the effect on the parietal lobe is accounted for by G?
I guess theoretically somebody could do that. We weren't really planning on looking at that for this study, but maybe it would be a good idea. I'm not really sure, but yeah, thanks for the tip there.
Just a comment. When I saw Jurassic Park at the beginning, when there's that bit when it goes woof woof with a puddle, and you have that kind of shiver in your throat, in your back of your neck. Sorry, I kind of feel like that listening to your talk. It's outstanding, and it's wonderful to see someone begin to trace out pathways all the way from genes through to actual biology, because that's kind of where we want to go. And thank you for fantastic work.
Thank you. Yeah, this is the stuff of thoughts, as Steve Pinker would say.
Are there other questions? Yeah.
I saw a number of cool mechanisms come up, but calcium ion binding behavior is commonly implicated in schizophrenia. Is this related to schizophrenia?
Thank you, Alex.
Between this and schizophrenia? Interesting. Do you think? Small but significant. Okay, well, okay, yeah.
Other